All right, so this is going to be a continuation of our biological macromolecules. We covered fats. Next, we're going to move on to proteins, okay? In terms of those proteins, they're going to be made of amino acids. And in terms of amino acids, there are 20 different types of amino acids out there. You'll see in the PowerPoint material I gave you, I have a diagram with all 20 of those amino acids. Don't anyone panic. I am not on the test going to ask you to identify a tyrosine versus a phenylalanine, or a lysine versus an alanine. What I want you to see, though, is that those different amino acids kind of fall into different classes based on the functional groups that are attached to them, okay? So the functional groups attached to the amino acid can dictate whether or not that part of the protein is going to be hydrophilic or hydrophobic. So whether it's going to like water or not. And that's going to be important in terms of proteins overall and how they come together and how they fold, all right? Because in terms of those proteins, all right, we have the primary structure associated with the amino acids, okay? Those individual amino acids are going to be essentially how the order that those amino acids are linked together are what make up the primary level of organization of proteins, all right? So if we have a valine, say a threonine, a tyrosine, the order those amino acids are in, that's our primary structure, okay? We are also going to have, with proteins, secondary structure, all right? And that secondary structure is going to get to some certain types of folding and how that protein comes together, all right? And in terms of that secondary structure, we're going to have alpha helices and we're going to have beta sheets. You'll see it sometimes as beta pleated sheets. Either is fine, okay? So we've got our sequence, we've got the initial folding that happens. Our next layer, I'm going to spell this wrong because spelling is my weakness. We've got the tertiary structure, all right? That tertiary structure is going to be how that protein comes together in 3 dimensions, all right? Typically, the tertiary structure here is going to be important for activity, all right? So for our enzymes to function, they have to have the correct tertiary structure, all right? If they don't, those enzymes are likely to not be functional, all right? So those are the 3 layers that I'm going to have you worry about in microbiology. Know that there is a 4th category associated with protein organization. So beyond tertiary, then we've got quaternary, which means you've got multiple protein chains coming together, okay? Not quite so relevant with bacteria as it is in eukaryotic cells. So for our purposes, I want you to know what primary, secondary, tertiary is, all right? And in terms of those amino acids, they're going to have a set structure associated with them, all right? And in terms of that chemical structure, I'm going to draw that here, all right? And this is the worst thing it's going to get as far as chemical structures for me, at least ones that I will show you, all right? And in terms of that, I'm not going to ask you to draw individual amino acids. But I do think it's important you understand the components of an amino acid because that dictates some of the function, all right? You'll notice here I drew a nitrogen connected to a carbon, connected to another carbon, okay? Off of those main chemicals, or off of those main elements, we've got some functional groups that are attached to them, all right? In general, the N-terminal region of the amino acid is the amino part. That's because that nitrogen bound to the two hydrogens, that's an amine, all right? So that's an amino group. At the other end, we've got a carbon with a double bond to an oxygen and a hydrogen, and then a single bond between an oxygen and another hydrogen, okay? So in terms of that, what we've got there is going to be our carboxyl terminal end of our amino acid, okay? And in terms of that, the N-terminal, the C-terminal of the amino acid, these are going to be where the amino acids are going to link together, okay? You'll notice also in there that I've drawn an R group. That R group is kind of a placeholder, a general placeholder. Because in terms of that, you're going to have some functional group attached there, all right? And in terms of that, if we're talking about that functional group just being a hydrogen, then our amino acid would be a glycine, all right? If you've got a ring structure there, it could be a tyrosine, all right? It could be a phenylalanine. Whatever's attached there dictates what the name of the amino acid is, okay? So I want you to keep that in mind. And as far as our amino acids go, getting those connected together now, it's going to occur got a C, an OH, all right? Between the N-terminal of of our next amino acid and the C-terminus of our current amino acid, all right? And what you're going to have in there is the formation of a peptide bond, all right? And in terms of that peptide bond, what you're going to lose out of that peptide bond is going to be water, okay? So in terms of connecting those amino acids together, that has a specific name. That's known as a peptide bond, all right? A peptide bond is a type of covalent bond, all right? So keep that in mind. All right, thank you for your attention.